Welcome back to Lively Life Snippets. Today, let's throw back to university days in 2016 where I was required to write a 7,000 words dissertation for me to graduate my pharm course. I chose pharmaceuticals related topics as I would to gain some experience on laboratory-based research, and formulation science is relatively new compared to biology and chemistry which we learnt in secondary school. I was assigned the topic on polymer grafting, and after experimenting for a month in the laboratory, I wrote my dissertation with the title of Surface Functionalized Polydimethylsiloxane, PDMS, with ethylene glycol dicyclopentenyl ether acrylate-di ethylene glycol, methyl ether methacrylate, EGDEA-DMEM, Copolymer Grafts, synthesis, characterization, and bacterial attachment study. I plan to do a series of 5 videos, and this is the first episode. After the cover page, I combined plagiarism declaration and acknowledgement in one page in my dissertation. In this section, I thanked my staff and student supervisors as well as my partner in laboratory. I will separate the whole dissertation in 5 videos, with episode 1 featuring abstract, episode 2 featuring introduction, episode 3 featuring materials and methods, episode 4 featuring results and discussion, and final episode featuring conclusion, future work and references. Abstract has 4 subheadings. Background and purpose: medical devices related infections are prevalent due to pathogens forming biofilms on their surfaces. Medical devices are made of many types of biomaterial. Polydimethylsiloxane, PDMS, or commonly known as silicone, is commonly used as catheters in urinary tract or bloodstream and implants in joints, breast and so on because it is biocompatible and biodurable. Bactericidal strategies available aggravate microbial resistance problems. To circumvent this limitation, strategies that can resist bacterial attachment are being investigated. Our aim is to compare the ability of thiol functionalized PDMS surface to resist bacterial attachment between those with or without polyethylene glycol dicyclopentenyl ether acrylate-di ethylene glycol, methyl ether methacrylate, EGDEA-DMEM copolymer grafts. Another objective is to compare graft from graft to in terms of resistance to bacterial attachment. Experimental approach thiol groups were added to PDMS sheets and confirmed by time of flight secondary ion mass spectrometry, TOF-SIMS. For graft to approach, EGDEA-DMEM copolymer chains were synthesized by catalytic chain transfer polymerization. Samples were submitted for nuclear magnetic resonance, NMR, and gel permeation chromatography, GPC, analysis for characterization of reaction and copolymers. For graft from approach, EGDEA and DMEM monomers were used. To graft the copolymers chains and monomers to thiol functionalized PDMS, 1% width/V photoinitiator were added. PDMS sheets with a drop of monomers or copolymers solutions each was exposed to ultraviolet, UV, light and degassed with AR for 30 minutes. Rinsings removed ungrafted copolymers and monomers. Samples were analyzed using AT-after. Treated sheets were incubated with RPMI media inoculated with M. cherry expressing Pseudomonas aeruginosa POW1 for 3 days with 60 revolutions per minute at 37 op prior to confocal microscopy analysis. Key results: TOF-SIMS showed increased intensity and a peak at 32.98 meters/Z in images and spectra generated. Confirmed successful introduction of thiols onto PDMS surface. Percentage conversion was derived from integration of monomer and copolymer peaks in NMR spectra. This was used to plot a graph of polymerization kinetic, which suggested as polymerization time increased, more monomers were polymerized. AT-after results confirmed that no copolymer were grafted on control, which was thiol functionalized PDMS. As concentration of copolymer solution decreased, greater peak area were observed, hence, more copolymers were grafted. At 60% width/V, no copolymer were grafted as the peak areas were similar to control. Graft from approach had greater peak area than all graft to approach. Thiol functionalized PDMS with EGDEA-DMEM copolymer grafts had significantly lower bacterial percentage coverage area than those without EGDEA-DMEM copolymer grafts, P equals 0.007. Graft from approach showed significantly lower bacterial coverage than graft to approach, P equals 0.0007. Conclusions and implications: thiol functionalized PDMS with poly-EGDEA-DMEM grafts significantly resisted bacterial attachment than those without. Less bacteria attached to PDMS grafted using graft from approach than graft to approach. For graft to approach, lower concentration of copolymer solution was associated with greater resistance towards bacterial attachment. These findings hinted factors needed to be considered during optimization of coating which resisted bacterial attachment. In future, ability to resist attachment of different bacterial strains can be investigated because few bacterial species are usually isolated from biofilms, rather than single species. Feel free to refer to this list whenever needed. A to H on this slide. K to Z on this slide. End of episode 1. Click on episode 2 for introduction.